Hey, welcome to today's Machine Learning for Protein Engineering seminar. Today, I'm pleased to introduce Stephanie Wankowicz from UCSF. She will be talking about the latest work on automated multi-conformer model building for X-ray crystallography and Cryo-EM with the goal of uncovering protein ensembles. All right, Stephanie.
This is why you unmute yourself before it starts. Okay. Yeah, thank you so much for having me. My name is Stephanie Wankowicz. I am an assistant professional researcher at UCSF. And my work really is kind of multifaceted. I really am passionate about improving the modeling of protein structures, both from X-ray crystallography and cryo-EM. And that's what I'm going to focus on here today, and sort of how we can then use the information that we get from dynamic ensembles of structures to interpret function or apply it prospectively to ligand and protein design. And so I hope, by the end of today that I can really convince you that I think for the next sort of breakthrough, the next steps in protein engineering and any sort of protein design is really thinking about a protein from an ensemble point of view. And so what I mean by that is, the many conformations that a protein can have, which often elicit function. And so with that, I'm going to get started and sort of emphasize what I was just saying, that proteins are dynamic molecules. They're constantly fluctuating between many different conformations. They're constantly fluctuating to both access states that allow them to do function, but also the fluctuations actually help stabilize them. And so even if we wanted to design a protein that didn't do any function just to have them stable, they need to be able to access multiple different states. This is important energetically. But importantly, when we think about almost any sort of function that the protein is doing, whether this is ligand binding, whether this is sort of catalysis, whether this is anything else, we have to be thinking about protein dynamics. And proteins can be defined by their energy landscape. So here is an energy landscape with the x-axis being accessible conformations. So these are the different states that the protein accesses. And on the y-axis is an energy. So higher energy indicates higher energy states. Lower energy is lower energy states. And in unfolded proteins can access many different states. So they really sit sort of up above at these high energy states, but they're wide. The energy landscape is wide. But when we think about sort of the structure that they have, it's just this gobbledygook of amino acids. They have no sort of structure. For the most part, they can't do any function. But they can access many states. And upon folding, proteins tend to lose access to many of these states. But notice that this is not a single point, right? This landscape still has a significant amount of width, indicating that proteins within their native ensemble are still accessing many states. And so we can think about this when we think about sort of the dynamics that the protein has, right? It's accessing and it's moving between these many different states. And so to be able to think about sort of how we are modeling proteins, how we're designing proteins, how we think about proteins, we have to be thinking about them moving between these states. And so when we think about sort of protein structure prediction, while, I think I'm going to be, fairly critical on the protein structure prediction side today, it's pretty amazing that we're even having this conversation, right? The fact that we sort of started to make massive inroads on sort of the protein structure prediction. But to me, I see this as a protein single structure prediction. So we can see that this is from the original AlphaFold paper, that the tracing between experimental structure and the AlphaFold predicted structure is great. Like it's the backbone tracing beyond it is unbelievable. And this can be said for a lot of proteins. But this static single structure of a protein isn't representing what a protein is doing. And so some really great data has come from TJ Lane, where he looked at all of the different conformations of this protein, the main protease of SARS-CoV-2. And so here in blue are all of the different structures that we have from this protease. And in purple, we have the alpha AlphaFold model. And so you can see that within the structures, we see a lot of dynamics. And this isn't even, a hugely dynamic molecule, but we can see a fair amount of dynamics and differences between the structures. And the AlphaFold is just to model is just really capturing one of these. And we can also look at that when we look at the RMSD between PDB to PDB. So this is looking at, two different structures and saying how different are they in the backbone. But we can also look at it between every single structure that we have in the AlphaFold model. And so in blue are these PDB to PDB pairs, and in this yellow is the AlphaFold to PDB. And so first you can kind of see that there is quite a distribution of the different conformations that a protein has in PDB to PDB. And this really indicates, the structural ensemble that this protein takes on, with each of these structures sort of capturing a different one. But then the difference between the AlphaFold to PDB, it's not really capturing like the range of this, right? It's a single model, so it's not capturing any of the ensemble. And the other thing is that the AlphaFold is not really sitting in the middle. And then when we also look at if we include ligands, so now we have a ligand bound structure, we can see that the distribution of the PDB to PDB is very different, right? It's now just has a single mode in it instead of a bimodal distribution. And so we can see that ligands tend to change the ensemble, the structural ensemble. But the AlphaFold model is not really picking up on that. And so if we then focus on sort of a very classic structure that goes between two different states, and these states being incredibly important, we can think about hemoglobin. So hemoglobin goes between the T state, the tense state, and the relaxed state. And this is its difference of being able to bind to oxygen. And so you can see that in these two structures that, in there's a narrowing and quite a conformational change. But if we look at sort of the AlphaFold 2 predictions of hemoglobin, it's kind of sitting right in between these two states. So we have structures of both the T state and the R state. When we look at this AlphaFold 2, when we look at both the PCA of it, but also when we look at the structures of it, it's just kind of like sitting in between. And so it's not really giving us information about these multiple different conformational states. And so people have really turned to how can we toggle the information that we feed into an AlphaFold 2 model to do this. And I'd say that the most popular, although there are many that I'm not going to be talking about today, of being able to do this is toggling the MSA, or the multiple sequence alignments, to this. And so the idea being that if we change this MSA slightly, if we reduce it, if we restrict it, that we can now, instead of just getting this single conformation, as seen sort of at the top here, now we might be able to get multiple conformations. And so this particular example is looking at kinases, which is, again, something that we know sits in multiple different conformations. We have sort of an inactive and an active structure, and many, many in between. And so what we can see is that, if we then sort of apply some of these metrics, that the AlphaFold 2 ensemble can now start to capture some of these. But it's very hard to tell, we have a lot of information about kinase and kinase structure and the ensembles that it takes on, but it's very hard to tell from this ensemble here exactly like what is going on. Is it just it's not predicting well? Is it that there is conformational heterogeneity here, or differences in the conformation? Or is it just kind of like capturing something in the middle? And so to be able to apply this to novel proteins that we might not have information about is very, very tricky. And so, my work involves sort of the fact that, we want to look at this whole distribution of states, which that, ensemble was predicting, right? Or, we could imagine that it's capturing all these different states, but we also want to know, what exactly is the difference between these two states, between these low energy wells. And so if we think about sort of protein design, we can also think about this in the same way where, originally protein design is very focused on matching to backbone. So this is from the hallucination paper where, it had this really great, in this incredibly static structure. So this kind of rock of a protein, but it had a pretty great RMSD to the structural model. But even when we start to look at side chains, which is sort of like my bread and butter, so beyond backbone changes, but side chain changes, we can see that there's definitely starting to see more of a difference. And so, I think talking about the fact that, my work really involves sort of sidechain conformational heterogeneity, I was super excited when, we saw the protein design moving towards the all atom. And so the fact that, we're not just focusing on backbone anymore, but we're focusing on different conformations of side chains involving, things like small ligands or post-translational modifications or metals, understanding sort of exactly how side chains are sitting is incredibly important. But once again, this is not really focusing on a conformational ensemble. So it's getting us closer to kind of those details. But for a protein to be able to bind a ligand, it has to go between certain conformational states. It often changes what's called the conformational distribution, or all of the conformational states that we're capturing in this energy landscape. And current approaches aren't really doing this. And so, I don't think that anyone is to blame in the protein prediction side. It's really my hypothesis is that protein prediction models need better training data. The fact that protein predictions don't exist with ensembles is because it's training on information from the PDB that is almost all static structures. So most of the PDB exists in a single conformation. And so we can look at this when we look at sort of the distribution of what we call alt locks. Alt locks is the way that the PDB enumerates different conformations of backbones or side chains within a single model. And so here on the y-axis is the percentage of alt locks. And then you can see, the percentage of structures. And then you can see that the number of alternative conformations per structure on the x-axis. And so, a bulk of the structures have no alt locks, and even up to the bulk of them are having under 10 alt locks. And so what this is really telling us is that the majority of the structures that exist in the protein data bank are single structures. And beyond that, oftentimes, if we want to get something that we understand the structure of, and most tools, they actually just even erase the alt locks. But I hope I can convince you today that alternative conformations of proteins are incredibly important, and it's really the work that we've been focusing on. So why, if you have a structure, how can you actually encode? Like, why should we be encoding multiple conformations of proteins? And this is really because X-ray crystallography and cryo-EM capture proteins in an ensemble of conformations. So here I'm just showing a single side chain, but it's representative of the entire protein. And X-ray crystallography and cryo-EM both get information. The experimental data that they collect is information from not just one copy of the protein, but it's thousands to up to billions of copies of the protein. And by capturing these conformations of the protein, we're actually capturing the protein in multiple different states. So even within a single fold of the protein, which is what we usually get from X-ray crystallography or cryo-EM, side chains still have the ability to fluctuate and keep that same stable fold. And these fluctuations are incredibly important. It is the fluctuation of a side chain is often what allows a ligand to bind. It's often what allows a metal to bind. It's often what interacts with water molecules, what interacts with post-translational modifications. Backbone or larger movements of the protein are also important, but side chains and these small fluctuations are often what can dictate a lot of function. But, things are not usually modeled about these ensemble of conformations, and this is not because structural biologists are lazy, but it's really because all of this information then gets averaged and squished into this data structure called electron density. So this electron density has all the information of the structural ensemble of these side chains and proteins, but it also has information with like tons of experimental noise. It also has information about solvents, about ligands, about anything else that's occurring in the experiment. And so to be able to understand sort of the signal that comes from the structural ensemble and peel that away from the noise of the experimental noise is incredibly difficult. But if we think about sort of the implications of this, if we have the same residue, but we have two different perturbations, let's say a ligand bound versus not a ligand bound, it would result in basically a different distribution of conformations. So you can see at the top, you have a much wider distribution of conformations that the side chain takes on compared to the other one. And that's incredibly important for a function, right? That's telling us sort of the width of this energy landscape. But if you model this in as sort of a static or single structure, as most of the PDB exists, you're not capturing any of this information. You're just capturing sort of this averaged view of a single conformation. And this is not telling us anything about the conformational ensemble. And this is really what exists in the PDB, and it's what is sort of the PDB is trained off of. And so what we aim to do is be able to capture the underlying conformational heterogeneity that exists from these multiple conformations that we can capture in structural ensembles and be able to capture that in our models. And so there are kind of two main ways that people have gone about doing this. There are ensemble structures, which is represented on the left, this purple. And then we have what are called multiconformer models. And so these are just trade-offs and differences of different ways to sort of encode the conformational heterogeneity. Ensemble structures are trying to explain all of the electron density that you have by putting in many, many, many conformations, oftentimes up to 50 to 100 different models that are explaining it, versus multi-conformer models are trying to do this in a much more parsimonious way. So by modeling in sort of the major conformations that a side chain might have or a backbone might have, but we're, reducing the number of parameters that we have in our model. And so my work really focuses on how can we automatically build these multi-conformer models in. I explained that it's incredibly difficult. This data structure of electron density is very difficult to work with, and it's very difficult to detect the signal compared to the noise. And so the way that we go about this is that we try to sort of automatically model a lot of this in, relying on optimization algorithms to improve the modeling between the experimental data and the models. And so I'm really briefly going to take you through our algorithm that we call qFit. And so qFit has been developed in the Fraser lab for over almost 15 years now. And we have a lot of really cool new directions that we're taking it. But I'm just going to very briefly walk you through what it does, and then we'll go through kind of step by step a little bit more. So what qFit does is that we take in a model that is pretty well solved. So you have to put in backbones, you have to approximately place side chains where you think it would go. And then we can rely on our sampling and scoring to be able to fit this in. So we first kind of start with some backbone sampling, and we get all of the potential conformations. And then we use this a mixed quadratic programming optimization algorithm to decide what is the best fit between all of the different conformations of the backbone and the experimental density. And then we do the same thing with side chains and sampling it around. And then we can place multiple side chains if supported by the density. And so this placing of the multiple side chains is where we get the name of multi-conformer ensemble. We're placing multiple conformations of the protein as supported by the data. And finally, because this is done on a residue by residue basis, we need to kind of put the protein back together, and we have to do what's called a labeling step. And so this is really looking at clashes across the protein and making sure that the protein is labeled properly. So breaking this down a little bit more, we first start with backbone sampling. And so we really do backbone sampling along the sort of up and down coordinate, so along the the alpha carbon, beta carbon portion. And this is really because, there's a lot less movement sort of side to side. You have restrictions due to the side chains being there. And then the question is, what is the best model to map fit? And so to ask this based off of all of our different samples of the side chains, what we do is we rely on quadratic programming. And so quadratic programming is defining both our model, our best map fit to model. And so this is defined by this minimization term, which we're looking at the observed density, this compared to a weighted calculated density, which is coming from our model. And so we allow the algorithm to potentially choose multiple conformations if it thinks that that is what is best explaining the electron, the experimental data that's underlying it. And we have a couple of requirements. We require that the minimum weight is a certain amount, and we also require that the sum of the weights is between 0 and 1. And then we follow this up with a mixed integer quadratic programming step. And this has multiple additional restraints on it to really choose the best up to 5 different conformations to fit the backbone. And so taking in each backbone conformation, we then start to sample sort of up the side chain. So we do this for every single chi angle or dihedral angle that exists up the side chain. And so by taking every single potential best backbone conformation, we then do exhaustive sampling, and we continue up this chi chain, the chi chain, until we built out the entire residue. And then at every single stage, we're doing this optimization algorithm. And finally, once we've built out the entire side chain, we now do the mixed integer quadratic programming step, but we also include a Bayesian information criterion step. And so this is really the fact that, the algorithm is built to be able to best explain the electron density. We tell it that it can fit up to 5 models, 5 different conformations. And that's because a lot of times there are times where 5 different conformations are needed to explain the very, very complex signal that we have in the underlying structure. But oftentimes, you give the model, the ability to model in 5 different conformations, it oftentimes takes us. And so this is just punishing the model based off of this K parameter by the number of atoms and the number of conformations that exist in the model. And so from that, we're now able to create this multi-conformer residue model, where we have both the alternative conformations modeled in. This gives us information about the coordinate, so exactly where each conformation is sitting. It gives us information about the occupancy of each conformation. So is alternative conformer A existing in what we estimate to be 60% of the copies of the protein and 20% of the copies of the protein? It gives us out information on that. And it also gives us out information on B factors, which is a metric of sort of noise and harmonic motion about each conformation. And so now that we have this sort of on a residue by residue basis, our job is now we need to put the protein back together. And so what we look at is we look at stretches of multi-conformer residues. We require, at this point, we're really looking at sort of backbone and making sure that we can stitch that protein back together. So we look for single conformer models, particularly of the backbone. And then we take everything sort of in between. We look at every single combination of the conformers, and we use, again, the same optimization algorithm with the BIC criteria to identify which explains best, removing different conformations as needed. And then from there, now the backbone is all set up, but what we finally finish this with is a simulated annealing step, which determines this appropriate labeling. So you can see here what protein models have is that if you have an alternative conformer A and an alternative conformer B, if you have alternative conformer B and B, they can see each other in the model. They can clash. They can have they physically cannot both fit in that space. But if you have an alternative conformer A on one side and an alternative conformer B on the other side, they now cannot see each other. And so as long as their occupancy are not adding up to more than one, meaning that, they're existing at the same time, they're allowed to take up the same space. And this allows us to sort of get this understanding of exactly how the different conformations of the proteins are fitting together. And so finally, we have this full multi-conformer model. And so the protein is now modeled in multiple different conformations, as you can see from the side chains here, as supported by the density. And so what we wanted to do to sort of really assess this model and understand, are we creating, our goal was really to create improved structural models. We wanted to explain the experimental data that we had underlying here. And so to do this, what we did was we took a set of very high resolution X-ray crystallography structures. And we asked, if we look at this metric of Rfree, which is explaining it is a validation metric that looks at left out experimental data and says, how well does your model fit into this experimental data? If we look at what is the deposited Rfree on the x-axis, so that's what exists in the PDB right now. And then if we remodel each of these proteins using qFit on the y-axis, what is their difference? How well is our model now fitting into the experimental data? And what we observed is about 80% of structures really improve. So anything below this red line indicates improved models based off of qFit. And so this really gave us a lot of confidence that we are actually capturing this underlying, the underlying data. And I think, regardless of whether you're, what kind of science or what kind of engineering that you're in, our goal is to always be creating models that are sort of best explaining the data that we have in whatever capacity you do that. And what this is really telling us is that by modeling in structural ensembles, we are actually explaining the experimental data that we have much better. And our data is trying to tell us that, it exists in this structural ensemble. This is what the protein exists in. And by not modeling this in this way, modeling the structural ensemble, we're really leaving a lot of experimental data on the board. And, if we think about this sort of one step further, right, we're leaving the fact that most algorithms, protein prediction algorithms are using just these models to then understand protein function, to understand the structure of proteins, to understand all of these different metrics. If we're not encoding this structural ensemble into our models, we're not actually explaining the structural ensemble of proteins. And so to visually see sort of like what actually does qFit able to detect, it's able to find sort of what we call hidden alternative conformations in high resolution X-ray structures. So here we're looking at the experimental data in this mesh, and then you see sort of red and green blobs. And the green blobs are indicating that our experimental data is telling us that that the model is not correctly explaining this experimental data, and the red blobs are saying your model is over explaining this experimental data. And so if we compare this to the qFit model, we now can see that there are multiple conformations of many of these residues, but also we've significantly reduced the amount of sort of over or under explained experimental data. And so by being able to model these in multiple conformations of, what is actually underlying and making up this experimental data, we now can explain it a lot more. And so our question was, okay, so we're explaining sort of the experimental data, but are we making physically realistic models? And so what we wanted to look at is, what does the geometry of our models look like? And we examined bond angles and bond lengths, and we compared this to the idealized bond angles or bond lengths as determined, and we saw that qFit models are much closer to the ideal bond and length. And so if we think about this, if we have a structure, if we have experimental data, it comes from an ensemble of conformations. And then we kind of try to just squeeze in, we try to push in one explanation of this conformational ensemble. Well, we're often going to end up straining the model, which results oftentimes in differences of bond lengths and bond angles. But if instead, if we model in the conformations into multiple different states, as the experimental data actually says, we're going to get much less flawed models. We're going to get models that are more realistic energetically. And that's because, right, this underlying data that we have is coming from proteins that are not at high energy states. They're at very low energy states. And by modeling in the multiple conformations that the protein takes on, we're not modeling in side chains in these sort of strained or high energy states. And so all of the data that I've shown you thus far is really looking at high resolution X-ray crystallography structures, but we also can do this in cryo-EM. And so cryo-EM is able to find hidden alternative conformations in, again, high resolution cryo-EM structures. But we can see that, the same sort of problem of the lack of modeling in conformational ensembles exists here. And although there are many other ways that we can model in larger conformational ensembles, it's very, very important to also pay attention to sort of these very delicate sidechain and backbone conformations. And we're really excited for the fact that in cryo-EM, we don't have what's called the phase problem in X-ray crystallography, which is just a way that the map is adjusted based off of the model. And so we think that we can push this resolution even more to look at structures that are at a resolution of 3-4 angstroms. And so if we now go back to sort of our different perturbations results in the differences in conformational state distribution. So again, looking at the different states of this one side chain that would underlie exactly what our experimental data looks like. If we compare sort of modeling this in as a single conformer state versus a multi-conformer state, we now can explain much more of this conformational state distribution. And we, now much better can, if we look at this bottom, maybe start to make some hypothesis, maybe start to extract some really critical information about exactly what these different conformations are doing and how this perturbation is changing the protein. And so, my hypothesis is that, the AlphaFold 3, we'll call it, or, maybe Rosetta Fold 3, or, your Fold 3, the next breakthrough is really going to require conformational heterogeneity. It's really going to require us to understand exactly what are the different conformations that a protein has and how is that impacting function. And without sort of getting there, then we're never going to fully be able to predict things that are impacting protein structure. And so I showed you, some examples of how conformational heterogeneity is very common in experimental data, but it's often unmodeled in structures. And the other piece that really plays a role in this is what is called compositional heterogeneity. So compositional heterogeneity is the fact that you can have different small molecules or peptides or even entire domains of structures that exist, but only in some copies of the protein that you're getting the data from. So if you imagine that these are all different copies of the protein in gray, you only actually have a ligand bound, in this case, in 3 of them. But then you get this ensemble average through the electron density. And the question is how do you separate out now this ligand bound conformation and the apo conformation? And this becomes even more complex because oftentimes, as I said, ligand binding can change the conformational heterogeneity. So now you sort of have this compositional and this conformational heterogeneity sort of mixed up. And I think for those who are, experimentalists, one of the really, really exciting things on how to get conformational and compositional heterogeneity moving forward is time-resolved X-ray crystallography or cryo-EM, or different perturbations that we actually can test in the protein. And so being able to actually capture all of this information requires us to think about how are we actually encoding this? What are the data structures? So not the structures of the protein, but the actual sort of models and how they're structured and how we're actually capturing this information. And so, why can't AlphaFold, why don't I think that AlphaFold 3 could take place today? And this is really, again, for the encoding of the structural heterogeneity. So if we have sort of two examples where you have conformational heterogeneity, you can think about this in a single side chain having two different conformations, or you can think about this in sort of a loop movement or a backbone movement of having two different conformations. Capturing this and encoding this in our PDB models is pretty poor. So this is the historical PDB format. And for those of you who are not familiar, the historical PDB format is actually going to be retired, I think probably in December. We are moving to the MMSIF/PDBX format. This is because we are running out of certain encodings of this structure. So this structure historically comes from punch cards. And I think that, anyone on this call can agree that any data structure that exists due to punch cards should probably not exist anymore, but this still does, at least until December. And so it's an 80-character line, and you are really defined by the different sections defining, different certain characters 1 through 6, defining whether this is what's called an atom or a het atom, so a ligand, a water, something that is not protein. And then there's like different information about the structure. But if we were to think about our conformational ensemble, how this is encoded is in what is right here, this AB next to the leucine information, which is called the alt-conf, the alternative conformation of this protein. And so this right now is how, if you were to run a qFit model, how this would be outputted, right? You would have multiple conformations of this leucine, and you could have it in an ARB conformation. But as I said, the historical PDB format is being retired, and we're moving to what's called this MMSIF or PDBX format. And this format is a dictionary-based format. So yes, we're moving into 21st century computing. And the fact that one, it is incredibly standardized, but it's also very extensible. And so it has the ability to encode a lot more information into these models. And so the proposal that I sort of have that we're trying to argue about with the PDB right now is that the MMSIF needs some amending to be able to capture the link between the conformational heterogeneity that exists in our experimental data and that's not captured in the models. So currently, the way that MMSIF is capturing the conformational heterogeneity is in the exact same way as in the historical PDB with this alt lock. But you could now imagine that you have something like you have two different conformations, but now let's say instead of in the loop out, you now in the loop instead of one conformation in the loop out and the loop in, you now have multiple conformations in this loop in. By linking this information through this dictionary, you now can get information on, exactly this atom. You can see that it is apo, so it does not have a ligand bound. It has two different sort of loop conformations, but then within one loop conformation, it has multiple additional conformations of A and B. And so you're starting to get sort of this layering of conformational heterogeneity. How does, if you think about this on sort of a large cryo-EM scale, right, how does the lid motion of a single cryo-EM conformation now interact with sort of the changes of conformational heterogeneity in the side chain or backbone level? That is completely uncaptured. And so if we think about this also with compositional heterogeneity, which is almost exclusively captured in the same alt-lock conformation, which makes it very confusing because it's two completely different things, but they're captured in the exact same way. You can now have a separate column, as seen here, of this bound versus unbound compositional heterogeneity. And if we get even more complex, right, and now we have a area where you have the structure, the ligand that is bound, some of the time, but then you have conformational heterogeneity above it. Now, because we're capturing the conformational and the compositional heterogeneity in separate ways, they have separate entities in the dictionary, we're able to capture a lot more of this ensemble. And so by encoding this in different ways, I really think that we can improve sort of our training for this, right? So not only are we sort of building out methods to be able to automatically model this, which will improve training, but we also are trying to really push on how can we encode this information, which is going to be critical, again, for really understanding how is conformational heterogeneity existing across the protein. Is this a loop movement? Is this a side chain movement? Is this an entire domain movement? And so with the time that I have left, I just want to briefly take you through sort of the applications of this, the applications of qFit, but the applications of conformational heterogeneity, and why I think it's so important that we include this information, not just on protein structure, but also thinking about it in protein function. So my work in kind of biology side of my work involves the fact that protein dynamics contribute to the entropy of free energy. So the entropy is determining the number of states that exist in a system. So the more states that you have, the more entropy that you have. And so protein dynamics, by moving between these different states, increases the amount of entropy. And so if we think about this on a protein folding landscape, there is a large amount of entropy at the top when proteins are unfolded, but when they become folded, they decrease their entropy, but they still have a significant amount of entropy. And so if you have an increased protein dynamics, you can have increased entropy. And so biologically, why is entropy important? Well, I guess physics underlies biology, but it all comes back to this equation called the Gibbs free energy equation. So the Gibbs free energy equation dictates any sort of protein function that you have. This delta G is the Gibbs free energy, and it's made up of a delta H, the enthalpic portion, but it's also made up of the delta S, the entropic portion. And so my work really focuses on how can we better estimate this entropic portion, which we are horrible at doing currently. And it is likely severely contributing to the fact that we really have poor estimates of delta G when we're estimating a ligand binding, when we're estimating mutations, when we're estimating any sort of other perturbation to the protein. And so a significant amount of protein conformational entropy actually comes from side chains. And this information we know from NMR, but it is the fact that side chains tend to access multiple different states within a single fold. And so if you think about all of the different side chains across the protein and the fact that they're able to access many different states, and the number of states, the number of different combinations between the side chains contributes to the amount of entropy, this is contributing a significant amount of entropy. And so my question that I really wanted to ask is, what is the role that conformational entropy plays in ligand binding? And so this is an MD simulation that comes from D.E. Shaw from their Anton computer, and it's of the Src kinase and its inhibitor dasatinib binding. And you can see now dasatinib is bound, but the protein doesn't stop moving. And so this is giving us really key information that the difference between the conformational entropy, the amount of states that it's able to access in the bound versus unbound state, can give us some really key information about entropy. But going back to sort of our static structures and why we need to improve this is that if we look at the structures of the Src kinase, we can see that there are differences in how the side chains are sitting. So maybe, with Rosetta all atom, we can start to capture some of this, but it gives us no information about the entropy. And so NMR has really provided some key insights into the impact of conformational entropy. And so here you're looking at red or blue dots that indicate the amount of disorder that different methyl groups have in in NMR, with red being those that become more disordered upon ligand binding, blue less disordered. And if we look at this if we look at the distribution between protein conformational entropy on the x-axis, what we were just looking at with the red or the blue kind of average, and the protein conformational entropy using ITC, and this is taking care of solvent and ligand entropy, we can see that there's this nice correlation. But what NMR really leaves out is the question of how is conformational entropy changing? And so if we think back to sort of a side chain, the structural implications of conformational entropy can come from many different forms. It can come from residues or side chains sitting in sort of one rotameric well, but they can also, different perturbations could impact the amount of conformational entropy by perturbing it into multiple different wells. And so the only way really to get this information is by modeling in structural ensembles, right? By using qFit and other tools that are able to model in these structural ensembles, being able to really understand what entropy is doing. And so the question is, how does conformational entropy change between perturbations, sort of the width of this, but also what are the structural differences in conformation? And importantly, how does this relate to enthalpic interactions? And this would really be able to explain both of this delta H, the enthalpic interactions, but also the delta S. And so what we can do is we can take multi-conformer models and we can actually quantify the conformational entropy. So now, because the data that we have is, much better encoded, we can look at the harmonic disorder, so the distance between the two conformations, but we can also look at harmonic disorder, which is the disorder within a single conformation. And so now we have this information where we can get quantitative estimates of energy, and we can get information about structure, and can we get more information about the mechanisms of protein function. And so what we did was we compared 750 stringently matched unbound and bound pairs of high resolution X-ray structures. And they were required to have the exact sequence and nearly identical crystallographic properties, and asked what is the conformational entropy changes on a residue by residue basis. And we were able to look at this across all of our pairs of proteins. And what we observed is actually that conformational entropy on average doesn't change. But when we looked at this compared to a data set of unbound versus unbound, so same crystallographic, but no ligand bound, we can see that the distribution is much smaller. And so this really indicated to us that the signal that we are seeing in the bound versus unbound is real signal. So now we wanted to ask the question of what is happening spatially across the protein. And the first thing that we looked at was binding site residues. And this is often because when we think about sort of how protein flexibility changes upon ligand binding, we tend to think about sort of the protein clamping down on the ligand. It wants to kind of hold the ligand in place. And so what we observed is that, yes, binding site residues tend to lose conformational heterogeneity. They tend to become a lot more rigid upon binding. And so this led us with this model where you have an apoprotein of conformational heterogeneity, conformational entropy changing across the entire protein. And then you have a ligand come in and the binding site gets less flexible. But distant sites, we really had no information. They either, the entire thing became less flexible, or distant sites tended to compensate by increasing their entropy. And this is exactly what we saw. We saw that the amount that binding site residues decrease their conformational entropy, the amount that distal sites tended to increase their conformational heterogeneity. And so this left us with this model of, apo state, the protein is moving around, but then in the bound state, the proteins have some sort of compensation that they do. And this is critically important. This is likely evolved. This is likely something that medicinal chemistry has been taking advantage of, that we didn't know about it. But if we can capture this information and predict this information, this would be huge in understanding protein function and ligand binding. And so we wanted to next ask, moving towards sort of the information of ligand, how does the same ligand bound or a different ligand bound to the same protein impact conformational entropy? And so to do this, we turned to a kinase CDK2, and we asked in the different rows, the different inhibitors that we had bound, how does this change the conformational heterogeneity in every single residue, which is in the columns here? Red, more flexible, blue, less flexible. And we saw that, there are sort of patterns of conformational entropy. And what we saw is that many of these patterns actually map to known structural implications. So the activation loop is known to be a reservoir of entropy. It's known to be flexible upon many ligands binding. And this is exactly what our data read out. But we also saw that, there are kind of two outliers, and these ended up being type 2 kinase inhibitors, which don't increase the flexibility of the activation loop. But this also really got us excited because not beyond sort of known stuff of kinases, but we saw that ligands sort of have unique conformational entropy signatures. So even within the type 1 conformational entropy, type 1 inhibitors, we can see differences in conformational entropy. And so, we are really focused on, and my lab is really focused on how does chemical and genetic differences change cause changes in conformational entropy. And this is both looking at chemical and genetic. But to get there, we have to improve the tools and methods to model structural ensembles. And so we're really focused on moving towards differences in solvent and how this impacts structural, how we can model solvent in structural ensembles, how we can do this with ligands, and how we can incorporate sort of our very small structural ensemble movement with sort of larger movements that you can see in Cryo-EM. And so by doing this, we really think that we can understand sort of the impact the conformational entropy has in many other aspects of biology. And this includes binding affinity and catalysis, macromolecular machines, and mutations. And so with that, I'm going to conclude and really say that qFit can improve our models of experimental data and extract quantitative estimates of entropy. If we want to get anywhere of really understanding protein functions, we need to stop thinking about proteins as static. We need to start thinking about them as ensembles and how we can capture these ensembles. And also that conformational entropy can have this spatially reciprocal relationship with ligand binding. And none of this would be able to be extracted if we weren't able to look at the ensemble. But this is like critically important of thinking about all of the biology that exists in our experimental data. But we are just, by poorly modeling it, we're not extracting it. And so if we can increase and improve our modeling and reduce the burden on modelers by making a lot of this automated, that we can extract more of this information. And finally, that different ligands bound to the same protein have unique impacts on conformational entropy. And so with that, I'd like to thank the Fraser Lab, and I'd like to thank really the qFit team down here. They have been a fabulous group of people to work with, and I will take questions.
Thank you so much for the great talk. Feel free to leave questions in the chat. I can read out the questions. We have one from Natalia. At higher Rfree values, maybe from low resolution structures, the program does not seem to significantly improve the deposited Rfree values. Am I wrong? What about the program's performance at low resolution maps?
Yeah, so I don't think I actually mentioned this, but the majority of structures the structures that we looked at were all better than 2 angstroms. So our program really, especially in X-ray, relies on high resolution structures. And it also really relies on pretty well modeled structures. And this is because the program is actually doing it all in real space. It's not doing it in a reciprocal space. So we're working on ways how we can incorporate this reciprocal space now to try to bump up both the Rfree and the resolution, but it is definitely a limitation of the method currently.
Also, feel free to unmute if you have any questions as well.
Hi, Stephanie. Can you, so maybe I missed that. So the data you showed, were some structure, the Rfree got worse. Are these also two angstrom structures?
Yeah.
Okay, so do you have a sense for why they get worse? Yes.
Yeah.
Yeah.
We don't. Or I guess I'll tell you a couple of reasons, but I don't have, there's not one definitive answer. We know that the model tends to do really poorly if there are really like very flexible solvently exposed regions. And that's one of the reasons why we're focusing on solvent. We know that the program tends to not, our backbone sampling isn't fantastic. And so modeling that in is something that we're really working on. And then the other thing is sort of this compositional and conformational heterogeneity and sort of incorporating both of those. But like I said, I couldn't tell you, I couldn't point to a dot on that graph and be like, this is exactly the reason why. It just tends to be that there are some models that just tend to make it worse, and we cannot fully pinpoint a pathology of that.
Yeah. And I'm not sure, maybe I misunderstood this, but it sounded like you fit up to 5 conformations.
Yes.
In your optimization algorithm. Does increasing that make things worse, or what's the interplay between that parameter?
Yeah, increasing it tends to improve the Rfree, but then we really start to get into to a point. So like we tend to really get into a point where we are we just think that the model tends to become way more complex than it needs to be. And so that was sort of the decision that we made to limit it to up to 5 and to also really include that BIC. Our thoughts are that we want a pretty simplistic and parsimonious model. This allows also modelers to then go in and manually edit the qFit models pretty easily in Qt or other visualization softwares.
Okay. Thanks.
Thanks.
Okay, I guess if there are no more questions, thank you again, Stephanie, for an amazing talk. And thanks for everyone attending. We'll see you at the next seminar series.
Thank you.